So as we continue our discussion on human genetics, let's recap. We've understood what the study is all about, the problems, and how we deal with the problems in studying human genetics. We looked at recessive disorders and dominant disorders. Now we're going to switch gears again and look at a different type of human genetic disorder, and specifically we're going to label the next couple of flowcharts abnormalities in chromosome number. Okay, so this is another way we can have a genetic disorder in humans specifically. So we'll entitle the next flowchart just like how I said. It'll be entitled Abnormalities, because remember, we're always looking at the normal versus the abnormal, as our first flowchart indicated. Abnormalities in chromosome number, and this will be Roman numeral 1. I think there are about 2 or 3 of these flowcharts. So this first flowchart on abnormalities in chromosome number will be all about and learning the terms associated with chromosomal number abnormalities. So let's look at the normal. In a normal chromosome number, what we have is something known as a disomy state. Okay, a disomy means that we have a normal state of diploid cells. This is just the normal way most cells are represented. So the normal state of a diploid cell. And remember that diploid simply means 2n, 2 copies of each chromosome, normal state. Every single chromosome has 2 copies in this diploid cell. This is normal. Disomy is normal. Thus we have, just like how I said, 2 copies of each chromo, and that is going to be sort of the idea of our homologous pair. Okay, so we can say in parentheses these 2 copies are homologous to each other. Why do we have 2 copies? These 2 copies, one from mom, one from dad, they're not exactly alike, they're just homologous. Okay, same size, same shape, same genes. Okay, just different variations of those genes. All stuff you already know. So let's look at some differences. Let's look at some variations, some abnormalities. One abnormality that can happen is something known as nondisjunction. Okay, so this is different. This is deviation from the norm. So it's called nondisjunction. So think of the name. It says non, that means without, disjunction. Disjunction means like separating, not moving apart. Think about where this would happen in, let's say, cell division. When would we not have moving apart? And I'll let you think about that as we explain it. So we have 2 ways of nondisjunction. Hopefully you already got them in your head. One of those ways is the failure, let's say, so this is all about failure. Failure of 2 homologous chromosomes to separate, okay, again, separation, disjunction, same idea, in a separation event called anaphase 1. So that's the failure that happens here. There's an anaphase 1 failure. The 2 homologous chromosomes don't separate. And if there's a possibility for a failure in anaphase 1, there's also a possibility for a failure in anaphase 2. So we can also have the failure, but we have to change our terminology a little bit. Failure of 2, but they're no longer homologous chromosomes in anaphase 2. Remember, they are now sister chromatids. So let's say failure of 2 SC for sister chromatids to separate, and now their separation is happening in anaphase not 1 but anaphase 2. Hopefully you understand that from our meiosis lecture. Okay, so couple of implications, couple of results of these non-disjunction events are the following. What's going to happen is, if this has to happen, of course, these chromosome number abnormalities have to happen in what we consider the gametes. Okay, this all occurs in the gametes. All of these things have to occur in the gametes because the gametes are what are going to be passed on. If we have a chromosomal abnormality in a somal cell, in an autosomal cell, does the offspring care? Does anything happen to the offspring? No, it's not going to happen in autosomal cells, or it might happen in autosomal cells, but this will not change anything about what the offspring will get. The offspring gets a gamete from mom and a gamete from dad. So if one of the gametes underwent non-disjunction, you're going to have the following. You're going to have a gamete with either the following, with either 2 copies, okay, 2 copies for a chromosome. What I mean by that is simply you have n plus 1. a gamete has to become haploid or diploid through meiosis. It has to become haploid, and if it becomes haploid, it becomes N. So that would be normal. Normal would be just to be haploid, N. But what happens if this failure of separation involves you gaining one extra because they didn't separate? What if you gained one extra chromosome? That would be one consequence of non-disjunction. But there's another side to that story. What if you are the gamete that was splitting apart from this non-disjunction event? You actually are going to have no copies. Sometimes you either are going to end up with both of them because of the non-disjunction event, so you might have both copies, or you might end up with none of them whatsoever for this chromosome, because one side of that cell in meiosis got both, okay, because they were non-disjunction on that side, and you ended up with the short end of the stick, otherwise known as N-1. So those are our two consequences of non-disjunction. It's a very good idea to look at a video or an image of this in your textbook. It's a great way to understand non-disjunction, and then we'll complete our understanding of these terms by looking at one final term, a very good, I think, cross-cutting term that really gives us an idea of all of the ideas behind chromosome number abnormalities. It's called aneuploidy. Aneuploidy simply refers to as the presence of an abnormal chromosome number. Presence of abnormal, so something that's not normal, chromosome, so we'll say chromo number. Anything that is deviating from the norm. That's what aneuploidy, ploidy referring to diploid, okay, something different from the normal ploidy. Presence of abnormal. There's an abnormal chromosome number, whether it's less or more, we're going to figure out. If you have a greater, let's say a trisomy, okay, there's a possibility for two things, trisomy and monosomy. So you should be able to understand which is which and what each refers to. If disomy means diploid, means normal, trisomy would mean that at some situation, at one chromosome, let's say, there's actually 3 copies, trisomy, 3 copies of, let's say, particular chromosome. So particular, let me rewrite that, particular chromosome. Okay, there has to be just maybe chromosome 1, 2, 3, 4, whatever. There's 3 copies of it, so that's why it's called trisomy. And all the others, okay, are normal. All the others have 2 copies of, let's say, all others. That's what a trisomy event is, when only one particular chromosome, let's say chromosome 1, doesn't have 2 homologous copies, but actually has 3 total copies. In this situation, we consider the individual 2n plus 1. Okay? Why is there a plus 1? What would this individual be? Disomy individual would be just 2n, right? A trisomy individual is 2n plus 1 because there's one extra copy at a particular chromosome. That's why there's 2n plus 1. Monosomy, you should be able to do process of elimination. This would actually mean the fact that you lack something. This monosomic individual lacks one member, we'll say one member of a homologous pair. So we'll call it, just for right now, a chromo pair. So chromosomes travel in pairs, they're found in pairs, and in this situation, in this monosomic situation, what we have is 2n minus 1, okay? Monosomy, so there's a minus over here. It should be disomy, but now we're going from di to mono, we've lost one. We've gone mono meaning 1. So this is actually interesting, because if somebody is monosomic, this is really, really bad. So bad that these people, these individuals, actually don't even get to be born. Most of the time, they actually die in utero. So we'll say it's die, it's called N, okay, this is Latin right now, in utero, within the uterus, okay, within their, let's say, prenatal development. Because they actually can't survive at all with a lack. You can survive if you have extra, but it's a poor survival. We'll talk about that a little bit later. But you can't survive at all unless you have both copies, without 2 copies. And what do monosomic individuals have? They lack one member. This is very, very bad, and thus they cannot survive. An interesting idea behind aneuploidy, and it's actually of great interest to many politicians, actually, and many individuals that are about pro-life and pro-choice is the idea that for the most part, about 50% of all pregnancies, actually, of all pregnancies are naturally aborted before about 8 weeks, okay? Before around the 8-week mark because of what we consider chromosomal abnormalities, okay? This is a known fact to many of us, to many scientists, based off of recent research. So this plays a critical role in understanding the idea and the complexity of pregnancy and life itself, right over here. That 50% of all pregnancies are aborted naturally before 8 weeks because of things like aneuploidy. Most of the time it's because of monosomy. So this is our idea behind abnormalities in chromosome number. We'll continue this discussion and get a little bit more specific now that we've established our terms in the next video.